hardworking staff, and in particular Mary, who go through a lot of work to set this up. These are nice facilities, but I'm also thinking that it's only going to be another year or two that we're using these facilities. And then it'll be great because I'll have a chance to host you in our own new building, which is on Orange Grove, just above, parallel with the Norton Simon on the other side of the 210. And then it'll be really fun. So looking forward to that soon. And finally, I wanted to thank Peter, not only for organizing this particular session of Neuro-Ophthalmology, but I know that he showed a lot of kindness in choosing which subject to give me. He showed kindness because he knows I love talking about optic nerve, and particularly about Leber's, and also that he knew that it was a timely event to do, because a few studies on this have come out. And I think there's been more science in the last year or two, certainly in terms of the treatment of Leber's, than in all the preceding period. So it'll be fun talking to you a little bit about what that shows. And in fact, that means that I'm doing a bunch of clinical trials. So there's these groups, Genzyme and Edison and Stealth, that I'm working with, just so you know that I don't take any proprietary interests. My connection is scientific. So taking a little bit of a broader point of view first, mitochondrial optic neuropathy. It turns out the mitochondria are critical to the function of most tissues in our body, all tissues. But the canary in the coal mine is the optic nerve. It is particularly high in its metabolic rate, and the optic nerve is very often the first thing to be involved if something goes wrong with mitochondria. So there are a couple of genetic causes for that are specific to the optic nerve, and one of them is Leber's hereditary optic neuropathy. That's what we're going to talk about today, because there's a mutation in the mitochondrial DNA. If there were to be a mutation in the genome, in that which you can inherit from mother and father, then it's a dominant optic atrophy, and it's the OPA1 gene that's involved. Whereas in the mitochondrial DNA mutation, you have the world's simplest genetic pedigree. If the mother has it, she passes all the mitochondria onto the offspring. The kids will all be carriers. If the mother doesn't have it, nothing happens. No father can ever pass it along. So it's rather easy to figure out that there's a maternal inheritance pattern. By the way, this is probably a good time for me to introduce three terms to you. If there's a gene that's mutated in mitochondria, we call that person a carrier. They usually will see 20/20 all their lives. But if they become blind, we call them affected. And that process of going from carrier to affected, we call conversion. Now, I told you that the optic nerve is the canary in the coal mine, which means it's also involved in more serious mutations. These are two of the mildest mutations you can possibly have that affects your mitochondria, so it only affects the optic nerve. More serious mutations produce syndromes like these. This is a myopathy, encephalopathy, lactic acidosis, and stroke-like syndromes. These patients often, as children, start getting stroke-like events and getting very deteriorated, GI effects, cardiac effects, neurological effects. But the interesting thing is, while these people have more to worry about than outpatient ophthalmology, they're all going blind too. If you look at any of their optic nerves, they show optic atrophy. So this is basically optic nerve plus. Speaking of which, you may hear the syndrome of Leber's plus. Leber's plus is the sort of spectrum event where you go blind, but you, in the 1 or 2% of cases, develop some other neurological symptoms that may mimic one of these elements. Okay, so what does it look like in our world? And all mitochondrial optic neuropathies have these characteristics. Bilateral symmetric vision loss eventually. In Leber's, it may not start immediately bilaterally, but it will become so. Early loss of color vision, central visual field defects, and both of these for the same reasons, because you've knocked out the macular fibers to the optic nerve, the papullomacular bundle. And that's why initially in all these diseases you have temporal optic pallor, because they're coming in from the temporal side. Now, if the disease is bad enough, it only starts this way, progresses across, and can involve all the optic nerve eventually. So I got involved in Leber's about 35 years ago because I was actually thinking about it all the time for these three great mysteries of the disease. And these three mysteries, of course, are also going to educate us about what's going on. Mystery one is variable penetrance. I just told you, most patients are just carriers. If you're a carrier, you're 20/20 until you get your cataracts in old age. You're fine. And yet, you might have a brother who is affected and goes blind and is hand motion vision. There's very little in between. So how is it that some people get involved and become affected and others don't? Well, one reason is number two. which is that your odds of going blind are very gender specific. Depending upon the mutation type, but for the most common mutation, it's about 5 to 1. So that means that there's a 50% chance of going blind under certain circumstances as a man, but only about a 10% chance of going blind as a woman. And we will talk about that mystery. Extremely tissue specific. Optic nerve, more or less, is the only tissue involved in Leber's, and we'll talk about why. And this one only has been figured out, I think, most recently, which is it's a genetic degenerative disease, like Parkinson's or Alzheimer's. But in Parkinson's or Alzheimer's, you've got this steady drumbeat of deterioration that takes place over many years and decades, but not in Leber's. In Leber's, you're a carrier and you're fine until you're not. And going from carrier to affected might take place over a week or two. So it's a catastrophic event. All the fibers are dying pretty much at once. Now, in the case of Leber's, it's one eye, and then a few weeks later, the second eye. It's essentially always a bilateral disease. A little primer in what mitochondria are. You remember them from medical school as these little beans that make all your ATP. They sit in your cytoplasm and have multiple copies of the genome in them. They've only got 13 genes other than their own machinery for making protein, and they supply the ATP. But they also supply almost all the reactive oxygen species. While initially people thought that the disease was one of bioenergetics, you weren't getting enough ATP, I'm here to tell you no. It's because you're making too many reactive oxygen species. That's the reason you go blind. The simple, and I call this my slide of little lies because this isn't exactly true, idea is that you pass from one complex to another the electron, and as the electron moves across this way in the membrane, it produces a proton gradient by pumping protons up here. This proton gradient is exploded to make ATP. Just to show one of the little lies of that slide, it's not really true that complex 1 passes to complex 2 to 3. It's actually 1 and 2 in parallel that pass the electron onto 3. And that's technically important because this is where all the mutations of Leber's are, complex 1. Had the mutations been anywhere further down the line, they'd be amazingly more severe because they would have blocked it during the series process. But here it's in parallel, so you can move electrons from complex 2 and bypass the problem, which is also why there is really no ATP problem. It's a problem that this electron has to go somewhere, and where it goes is to produce reactive oxygen species. So fortunately, I've learned a lot about Leber's by being able to take advantage of the world's largest published pedigree in Leber's, which is in Brazil. And so we go down every year. Peter has come down with us, and we have a team of about 40 people that looks at the same patients year after year. So now we have about 20 years of longitudinal data on this enormous family. And the family has been fully developed to have 362 people going across 8 generations. It began with an immigration from Italy of this woman at age 16 who was being blindly married off to somebody in Brazil, and they've lived in this rural corner of Brazil ever since between these 3 cities. And this is the pedigree. We don't have to pay much attention with it. It's very, very extensive. The initial person who emigrated from Italy is this one right there. This little corner of the pedigree actually is still in Italy, and because all this work was done by exploring cemeteries and church records in Italy during his summer vacation for August, we have allowed the person who did this, Corelli, to give the name to the pedigree, and so he calls it the Soave Brazil pedigree because it began in the Soave region of Italy. We're going to talk a little bit about that carrier right there. Initially, we looked at things like gene linkage analysis. I'm not going to have time, or you don't have interest to go into that today. Environmental factors, and I'm going to just very briefly mention that the odds that the patients who go blind, which is this group here, smoke, is much greater than the background population. That they drink heavily is much greater than the background population. Now you might see with interest that the carriers are even lower than the background population. And that's probably phenomena statistics of ascertainment bias, because these carriers who smoked a lot were more likely to not be carriers anymore. They flipped over and went to this column and became affected. So very simply again, a slide of little lies, is that you've got these in parallel, complex 1 and complex 2 donating the electrons, and in Leber's you knock that out and you produce reactive oxygen species instead. And so to expand that model a little bit, I've got this drawing, which now appears apparently not always credited, and lots of journals and lots of book chapters about Leber's. But this is the retinal ganglion cell. This is the retinal nerve fiber layer. This is the lamina curvosa, and now the optic nerve, which carries it to the brain. And the slide illustrates the fact that mitochondria go where their work is needed. And their work is needed where there's no myelin. So they cluster in the nodes of Ranvier. Well, the world's largest node of Ranvier turns out to be this, the retinal nerve fiber layer, because we don't have any myelin in the eye. And so this explains in large measure why the optic nerve is the canary in the coal mine, because most of the work of an axon is where the saltatory conduction occurs, where you have to re-establish the sodium and potassium gradients. And so this is an enormous amount of work. And in fact, this is where a huge number of mitochondria normally are filling. So if complex 1 is blocked, you make reactive oxygen species. If you aggravate it by exogenous sources of reactive oxygen species, smoking and drinking, you get to a certain point where you produce so much that that the potential across the membrane of the mitochondria gets to be a certain amount. Normally it's minus 200. If it drops down to about minus 120, it opens these little pores, the mitochondrial permeability transition pores. They're gates, just like you've learned about potassium and sodium gates. It's a gate for this very big molecule, though, that slips out, and that's cytochrome C. The same cytochrome C that's involved in oxidative phosphorylation, and this event sets off apoptosis and kills the cell. And it's a mechanism that the cell normally uses for apoptosis, which in this case is getting a mistake, a false signal. It thinks the immune system that is normally surveying cells and saying, you're misbehaving, kill yourself, is telling it to do so through the signal of reactive oxygen species, but it's coming from this false source, and the cell dies. So, I'm going to concentrate first on asking the question of why is it so tissue specific, not just of the optic nerve, but specifically the papillomaculaire bundle. Remember the clinical symptoms of that. And to that, one of the studies that was done by one of our colleagues who came to Brazil, called Barboni, is he looked at the OCT, the retinal nerve fiber layer, and he looked at it by sector. And so, while normally we know that in the OCT, a month or two after you have the problems of vision loss, you have swelling of the fibers, because they're stuffed with mitochondria, by the way. And then eventually you have optic atrophy. He asked what happens section by section. And what he found was that initially the swelling was in the inferior temporal quadrant. It's probably the temporal quadrant more than we think, but the OCT is not very good with the temporal area, so we see it mostly inferior temporal. It swells first, it dies first. And then later, the last thing to go is superior nasal. It swells last, it dies last. So there's a wave that marches. Now, for those of you who are more inclined to knowing the anatomy of the optic nerve, this is striking because the smallest fibers in the optic nerve are these fibers. And the largest are these fibers. So it obviously suggests the idea that small is bad. So we looked at it histopathologically and confirmed that this is the wave that's going on. We have 17 different sections of optic nerves from, by the way, the patients in Brazil all are kind enough to sign consents and provide their eyes for autopsy when they die, and so we've had a lot of chances of looking at it. And I'm giving you here 4 pictures, which I've ordered from the mildest case we had to the most severe cases and 2 intermediate cases. But remember, these are not longitudinal. This is not the same optic nerve marching along. These are 4 people who died, and I'm lining them up also so the temporal is always on your left. And staining it with a stain that shows normal fibers. And what you see is there's a bite out of the temporal side that starts the process. And in some patients who are worse, a bigger bite, an even bigger bite, and in this worst case, just a few fibers preserved supranasally. So we got that wave again that was shown clinically. We can recapitulate it histopathologically. So at this point, it seemed reasonable to develop a math model for the whole disease. And the math model is this one. It says that the likelihood of fibers dying, this is the X, is due to the number of nodes, because that's where the work occurs, times the length of the node, the more membrane, the more you got to work, times the circumference at the node, more membrane again, divided by the number of mitochondria you got, and supplying the process. And fortunately, this can easily be reduced from the geometry to the algebra. This is length times 2 pi r. These things are 2 pi r. And the volume of a cylinder is length times pi r squared, and that's how much mitochondria you can stuff in there. And everything cancels out, leaving 2 over r. So what 2 over R means is big is better. The large radius of curvature is going to decrease the total risk factor. So we decided to test it. And I had, I think that you remember him, Billy Pan. I had a student who took a year off to do this. He took electron micrographs, and he counted all year long, hundreds of thousands of figures. This is 100,000 in just one optic nerve, and it happens to be a normal optic nerve, so forget Leber's for a second. And he created these nerve fiber spectra, and he did it for a 6x6 quadrant, throw out the 4 corners because they're off the nerve, so 32 sectors of the optic nerve. And what you find out is that there are more small fibers here in the infratemporal temporal side, and more of a shift to the right in fiber size there. So it's exactly what we thought would be the case. Then, of course, at this point after a year, and he says, I'm finished, we did a photograph from here at the same mag as a photograph from here, and you realize the difference is so great you can just see it. But I'm glad we got the numbers because you'll see why the numbers are better. You see how the fibers are smaller here, you can see in cross-section, high mag, same mag here, they're bigger there. So small here to large there, and there's a really good reason for that, by the way. Isn't nature wonderful? These have the shortest distance to go. They're going straight to the macula. These have the greatest distance to go. They have to go to the far nasal retina, which is the furthest from the optic nerve. The further something is, the longer it takes the message to get in. And if when you saw something, you got the message at different times because of where it was in your visual field, that'd be pretty disturbing. So to perfectly re-normalize, so all the timing occurs simultaneously, the longer distance fibers are larger, which means their conduction speed is faster. Beautifully balanced. So then Billy recapitulated this in a way that we could just optically see it. White is small fibers, black is dark, and the grayscale showed the size of the fibers. And I found one particular optic nerve that I thought remarkably matched it. I want to emphasize here, these numbers come from a normal optic nerve that Billy calculated. This comes from a patient with Leber's. So you can see that the math model really does fit nature quite nicely and answers the question not only why the optic nerve is vulnerable, but why the smallest fibers of the optic nerve are most vulnerable. So we've knocked off, I think, a few of the mysteries. We know that environmental factors like smoking and drinking affect penetrance. We know that tissue specificity makes sense. Neurons, optic nerves, small fibers. What about gender bias? I'm only going to show one slide on that because I don't want to waste too much time on it. But we found out that estrogen is protective by increasing the mitochondrial copy number. This is a graph that shows that in controls, mitochondrial copy number is about 200 per cell. If I further fractionate this into males and females, it's lower for males and a little higher for females. If I look at people who've gone blind, it's higher. But this is the most important one. If I look at people who did not go blind but had the mutation, it's higher still. Probably by increasing their copy number, they were able to protect themselves from becoming this group. And this copy number was even higher amongst the women and lower amongst the men. And it's because copy number is mediated by estradiol to the beta receptor. So, better to be a woman. So I think that explains very much the gender bias, and it allowed us to go back and do another study which hadn't been done before because people didn't have big enough numbers. And we asked, we plotted the number of conversions by age, and it turns out that for men, there's this big peak about age 20. But for women, it's a very small bump at age 20. But there's a second bump. Anybody want to guess the age? Age 50. So menopause puts them back at risk. Finally, this one has been the mystery that has gnawed at me until the last year or two. Why is it that you go from this, the presymptomatic stage, what the optic nerve looks like, with that swelling of the nerve fiber layer down here, through an acute phase, and then you get to the post, and why does it happen only over a period of a few weeks, so catastrophically? So I have to give credit here to Len Levine up in Montreal, who had access to some huge computers. And he came to us and he said, you remember that data from Billy? Can we have the raw data? And we're going to feed it into our computers, and we're going to do Monte Carlo simulations. But before I get to that, I want to explain the thinking that he and I had. So you remember how the atomic bomb works, nuclear fission works. You end up with a neutron banging into an unstable uranium-235, breaking it up, and releasing 3 more neutrons. Each one in turn breaking it up. And so you now have this critical pile blowing up because each neutron induces more production of neutrons in the next atom it hits. Now, how would you mitigate that you would use graphite rods, and that's what you do for a reactor, because a graphite rod will absorb the neutron and not emit any more. Well, this was our model, except instead of neutrons, we think in the optic nerve, it's reactive oxygen species. And instead of uranium-235, we think they're small axons. And when the reactive oxygen species reach a critical threshold, it causes that axon to die and produce that many more. And for a graphite rod, we think it's a large axon, because a large axon can absorb the reactive oxygen species and not die. Okay, it's a nice theory. How can you prove it? And here's the exciting thing. We took this data from Billy, re-entered it, and asked the computer to go through millions of iterations based on the probabilities of a small versus a large fiber going through these events. And we started off with the idea that the first fibers that died might have been on the nasal side, where the fibers are large. And I got the wrong picture. Okay, it came out of order. This is when it's on the temporal side, and it spreads, and it spreads, and it spreads all the way around. Sorry that I showed the wrong picture, but that's what the explosion event of the Monte Carlo simulation showed. And on the nasal side, it just fizzles out because there's a lot of large fibers to absorb it. What I wanted to do by reversing the order of these pictures, which I can, I think, do just by going through it, is to show this picture. And you notice that this is in a patient, there's a simulation of a patient who is normal. The Billy had counted had a normal optic nerve. This is a real Leber's patient, and this simulation produced everything as it curled around the edges and even came up through here, just like in the case of the Leber's patient. So with this supercomputer, I think we recapitulated the disease and explained how we get from the pre-symptomatic to the post-event like that. I now want to talk, let me go back a second, about treatment, because that's what Peter brought me here for. And the question is, this is the natural history of the disease, where you get from a small central scotoma to a big one, and then it tends to either flatten out, or we hope with treatments to improve a little bit. What can we do about it? And the first study was done many years ago, both by our group and by a group in England. Retrospectively, we did it, and prospectively, they did it. This is our study in a large number of patients, and this is theirs as part of the RODOS trial. And the answers turned out to be almost identical, so that they did reinforce each other very nicely. In the RODOS trial, they looked at almost 100 patients. They used a standard dose, placebo-controlled, all done properly, except for two things. One is they only looked for 6 months, and two is they used probably the wrong endpoint. But nonetheless, they did find some interesting things. Unfortunately, they didn't get the p-value they wanted because they picked the wrong endpoint, and they didn't get it because they looked too quickly. Turns out that even though this agent, idebenone, metabolically improves things in a Leber's optic nerve cell in a matter of minutes, the effect probably doesn't play out except for about 9 to 12 months later. And they didn't get far enough out to show the statistics they wanted there. They did do some other subgroup analyses, which obviously did not persuade the American FDA because they were cherry-picked, but they were not able to prove what they wanted to initially. This is one of the analyses that we did in our paper, which was in a large number of patients, and the most important thing is over a longer period of time. So at 6 months, which is when they finished the study, the two curves are touching. But the longer you go out, the better you see the effect of idebenone in protecting the death of cells. Now, it also turns out that this particular mutation works well. If you do 1444s, they get better on their own, and if you do 3460s, they never get better. So it worked out that we were able to look at the particular mutation that responds the most to the therapy. Now, the next 5 slides, I asked for from GenSight and had permission to use. I remodulated them to simplify them a little bit, but it talks about gene therapy for Leber's. And the idea is that you take a virus, an AAV2 virus, you attach to it the ND4 subunit, which is the one that's missing in the most common mutation for Leber's. We're only going to look at that one, the 11778. You inject it intravitreally, where it gets absorbed by the retinal ganglion cell, and this particular gene, the ND4, is made. Now, this has been done actually by different groups, including John Guy in Miami. What makes the French group that sponsors the trial that I'm going to talk about more powerful is they added to it a second element. They call it the MTS. It stands for a mitochondrial targeting sequence. This element has been shown to associate with the mitochondrial membrane. And they've been able to demonstrate that this drags this protein produced in the nucleus, because normally it's not. Normally this protein is made by the mitochondria for itself. So we're taking a workaround by putting in the nucleus, getting to make a protein conventionally, and then using this targeting sequence. Now I gotta tell you, one of the things that bothers me is the targeting sequence is for the surface of the mitochondria, and where it really ought to be is on the inside. But they say magic occurs and it gets inside, and for the moment we'll accept that. So there's 2 trials that have just been concluded. The one that they call rescue is inside of 6 months of vision loss, and they almost 40 patients. And the one that they call reverse is from 6 to 12 months. In both cases, it's a randomized placebo-controlled injection, so that one eye randomly chosen gets injected with the virus. The other eye gets pseudo-sham injection. That means that they just press the hub of the needle. But after you had a little subconj lidocaine, you can't tell the difference whether there's a needle or not. And so without putting the patient any additional risk, they don't know which eye got injected. We do the injections at Stein, which is the unmasked center. We do all the other work at Doheny. Yeah, they're unmasked, I'm masked. I don't know which eye got injected. The patient doesn't know which eye gets injected, and then we follow them through. Okay, what do the results show at 72 weeks, a year and a half out? Well, in terms of ganglion cell macular volume, the treated eyes maintained themselves, the untreated eyes lost a little bit. That difference is really tiny, so I would not know whether to call that significant or not, but statistically it is. Retinal nerve fiber layer. The treated eyes lost almost 2 microns of thickness. Remember, we're talking about 100 microns background. That's not a whole lot. The untreated eyes, about 4 microns. This 2 micron difference, again, I would not think would have great clinical significance, but statistically, yes, it is significant. And so those are the 2 big structural outcomes that are being used, and you've probably seen the press releases to claim great success for the gene therapy. I find this the most interesting display of the data. So this is the visual acuity in terms of how many ETRDS letters there are. And so the visual acuity following the injections, and this is, remember, on average, this is actually for the reverse. So this is probably about 9 or 10 months out. Patients already gone blind. They do the placebo injections in one and the real injections the other, and the patients who have the placebo do nothing for a little while and then slowly get better. This is the eye that is not getting the virus, and the eye that gets the virus more. So they're touting the fact that that's 3 lines of vision. That is impressive. But the other eye got 2.5 lines of vision. It's not that big a difference between them. And I'm further impressed by how parallel these 2 lines are, suggesting that if there is anything going on, it's right there. Or it happened randomly right there and just got preserved. You could decide later. But the effect is not statistically significant on this issue, so they're not allowed to call it a rescue or a visual acuity. But what they did get statistical significance was in contrast sensitivity. And if anything should impress you, it should be this one. These changes are minimally clinically significant, but they're definitely statistically significant. And with a little bit of data manipulation, they set a threshold, post hoc of course, that said that by this threshold, half the treated eyes and only a quarter of the untreated eyes showed a contrast increase. Now there's a little bit of data manipulation that I found very troublesome, which is why my name's not associated with these press releases. But nonetheless, we got a little bit of an effect. And they're about to go to phase 3. You're probably going to get letters from me asking you to refer these patients in. Phase 3, reflect trial. There's going to be bilateral injections. The patients guarantee that one eye that gets injected will be the real thing, and a 50-50 chance that the other one will be sham or the real thing. So now we're going to be getting more of injected eyes to compare against the uninjected ones. Okay, those are the gene therapy trials. I'm going to talk about one more trial very briefly. This is an eye drop made by stealth peptides. It produces a chemical that relates to cardiolipin. It has been shown in animal model to decrease reactive oxygen species. This chemical, cardiolipin, no, just skip that part in fact. This is the chemical, produces a cage, and it's called MTP-131. We just finished and closed our trial with not only 12 eyes of placebo control, but now we are finishing the open extension. And bottom line, if you look at the data, there's a minimal effect. In Humphrey visual fields, they got the statistical significance in terms of getting about 2 dBs of improvement. In contrast sensitivity, about the same as the gene therapy trial gave. Again, their way of manipulating the data is to say 10 of the 12 eyes showed improvements in either visual acuity or visual field. But what's interesting is that if you go back and find the data, which I had to dig for, 7 of 8 placebo eyes showed improvement. Both studies showed improvements in the placebo side. And this is, in fact, that raw data that shows that there is, this is the 1 year mark, a difference between the eyes that were not treated and the eyes that were treated. But look at what happens in the 6 months that follows with open extension. Both eyes are getting treated. You're getting an extra improvement and then the difference between them. The easiest way to interpret this doesn't make sense, which is that somehow injecting one eye benefits the other, that putting a drop in one eye benefits the other. There's no systemic explanation for why that would be the case. So there are now 4 new placebo-controlled trials that are pretty much closed, 3 on gene therapy and 1 by these eye drops by stealth. And I want to finish with telling you what I would advise. Given all this, and I've obviously had this issue come up on every week, what do I do with a new patient? I put them on Idibinone. The safety profile for Idibinone is the best. They can get their own Idibinone on the internet by, you go ahead and take a photograph, typing in the words Idibinone and either HBC or Relentless. They both sell them as food additives. It's legal for them to get it. It's illegal for you to prescribe it. Then I will talk to them about the clinical trials, knowing that the longer they're on Idibinone, probably the better, because they have to stop taking Idibinone if they go on the gene therapy trial. But fortunately, they do not have to if they go on the eye drops, so they get the benefit of both. And the reason I say the longer the better is I've done my own personal review of the data, which is not official, which says the patients that did best with gene therapy trial had the longest Idibinone pre-trial period and then had the injection on top of it, which makes sense. You might as well get a little bit of benefit from 2 directions. And as I mentioned, that they're getting tremendous press release. If you read this, it sounds like it's a great sea change, but I think that there's some exaggeration. It turns out that Idibinone and Epi-743, which is similar to Idibinone, have similar press releases. All of them work a little bit. And that's the group that I've taken to Brazil every year, for which all of these studies profited a great deal from. They're taking their vacation in rural Brazil. And I think, Peter, you're in that picture somewhere, but I can't find it right now. Thank you very much for listening to this.
We have time for 2 questions. Are there any questions for Dr.
Yes.
Sadun or any members of the front row?
Yeah, how frequently do you give the injections, or is it a one-time?
It's a one-time
how frequently
thing.
is the injection given?
One thing. The AAV2 virus carries the little gene in with it, drops it into the retinal ganglion cell, and the presumption is once incorporated, it's there forever. Now, this was based on some of the science that took place with the, as you know, there's Leber's congenital amaurosis, which has nothing to do with this Leber's, but which has also been treated with similar intravitreal injections. And after 1 year, they had a better way of measuring the responses. They did really well. And after 2 years, there's been a little bit of a decay. So there may be reasons, this has not been addressed yet, to say that there's competition between the newly introduced gene and the mutated gene that might favor the mutated gene over long periods of time, and you might need to re-inject. But up to now, nobody's done that.
Well, how does the gene get to all the mitochondria?
Well, that's the
question,
magic.
Alfredo,
That's
can you
the
repeat
magic?
the question, please?
Yeah. And the question was, since the gene gets into the cell, and now the protein product is made by the nucleus, but that's not the normal way that this particular product is made. It's made by the genetic machinery of mitochondria. How does it get to the mitochondria? And the company claims that their patent for MTS, a mitochondrial targeting sequence, is what it does. And they're right, at least for the first half. They have found a transporter system that the cell uses to move other things to the mitochondria that they hook it onto, and so it gets transported to the mitochondria. The magic that I find uncomfortable is that only brings it to the mitochondria. It doesn't explain to me how it gets inside of the mitochondria.
So in your rescue trial, you mentioned that there's an increase in contrast sensitivity, 50% in the patients that are treated, and that correlates with the thickness of the ganglion cells.
That's right.
You think about the ganglion cell layer, of course.
Yes. Well, there's 2 microns improvement in the retinal nerve fiber layer that comes from those ganglion cells. So that's the biggest structural I mean, it's a small structural advantage, but it's the most impressive. It correlates also with what you just said, which is that there's an increase in contrast sensitivity, and that's the most important functional advantage.
Thank you.
Thank you very much. So that brings us to the conclusion of the first half of our program. We're going to have a 25-minute break, and we'll reconvene at 10:30, please. Thank you very much.